The whole idea of these workshops and tutorials is to give you a good idea of how to use freely available tools, as well as some of the tools we've developed inside the platform. And the first three are really to do with like brain analysis and visualization. So we sort of went through ClearMap last week, and this week we're going to look at how to use BrainJ for things like confocal images and images from the slide scanner. And then next week we're going to look at using BrainRender and some open source tools for doing the visualization and rendering of some of these data sets, because in some of the publications we've used things like Imaris, but that can be incredibly tedious and time consuming. So actually using some of these open source tools can save you a lot of time and even give you a better result. When I joined the Zuckerman Institute back in 2017, we had a sort of custom slide scanning microscope that many of you may have seen. And this is a nice microscope. It has a low magnification, high numerical aperture objective, which allows us to sort of image with a large field of view, but still have bright images. And initially we were just scanning in whole slides. And so one of the first things was really to try and optimize this a little bit better. And so we developed some routines just to isolate the tissue sections and then image those using all the colors. And the resolution that we could get doing this is about 1 micron per pixel, which is more than enough to do basic cell quantification and also start to get a measure of projection density and things like that. And if you're interested in how to use this sort of segmentation and tools like that in elements, this is something we'll be talking about in a future workshop in 4 or 5 weeks from now. And just to show you like the images we can get from this microscope, you can see cells quite clearly, and we can see these areas of axons and dendrites, especially in virally labeled cell populations. And so we had this data, but when you look through the literature at the time, there was many papers where people had done this type of analysis, but the tools were not really shared properly or weren't working, or some work, but you needed to combine several of them, and it wasn't something that was very easily accessible. And so the idea was really to create something that was reproducible, high throughput, unbiased, but importantly, something that was also really accessible. So we wanted it to be something that like an undergraduate student visiting a lab could come in and actually use to get some usable data from brain scanned on the slide scanner or the confocal. And so with that in mind, we developed something in ImageJ or Fiji that combines all of these tools and is very easy to use, and it doesn't require any coding or anything like that to interact with. And we call this BrainJ. And so the general gist of it is we have images coming from the sort of slide scanner. You need to put the sections in an anterior to posterior order, but then we can sort of reconstruct that into a series of sections in this anterior to posterior order. And then we need to be able to pre-process those to clean them up and then register them serially with each other to have a cohesive 3D volume. And then we want to be able to detect the cells and axons and dendrites. We want to be able to do that in a way that's better than just doing simple segmentation. And so we use machine learning for that. And then we want to be able to align that to the Allen template brain so that we can start to do measurements inside the atlas. And so we do that sort of alignment, and then we can do things like looking at cell locations, the density heat maps, and then a whole lot of region-based analysis. So projection density, mean intensity, and a whole lot of things that especially the people in Rui Costa's lab and some of the other labs that I was working with early on, that these are things that they wanted to be able to do but couldn't do, in a way that without, either couldn't do or would require many days of manual work. And so these are the sort of things that we built into the pipeline. And so just to give you an idea of the first part of this, you have images that are coming from the slide scanner, and then we want to sort of be able to have them in a more cohesive volume. And so we do like a serial registration after cleaning up and already sort of preparing the tissue for this process. And this allows us to get a very good registration, even when the data can be quite messy coming from the slide scanner. And then importantly, because so many things can happen in the preparation of these samples, a lot of people are doing floating sections when they're labeling, and so that they need to be able to flip the sections. there's many sections, so things can go wrong. You need to be able to figure out, which sections need to be manually sort of checked or something like that. So the very first pass in the pipeline is to give you a montage view of what the brain looks like. And this lets you sort of see, okay, like, in this case, I can see that section 29 is actually flipped horizontally to the other sections. And so, it's very easy to just quickly go through and see, okay, these sections need to be flipped. This section, if this tissue is actually, I don't know if you can see my cursor right now, but the bottom right corner, that's fine. But if that tissue is actually touching the other brain section, that's something we'd have to quickly manually edit. And I'll show you how to do this during the tutorial. And once we've done that, we can then do this automated registration and then a 3D alignment to the Allen Brain Atlas. And then we also incorporated machine learning. So Ilastik is a fantastic tool for doing machine learning pixel segmentation. And I'm actually going to go through this briefly in the tutorial today, and we'll go through it in more detail in a later tutorial. But this is a really fantastic tool for segmentation when, very traditional sort of segmentation tools just aren't sufficient. And in our case, we trained it to detect both cell bodies and axons and dendrites, as well as removing background. And this gives us a really, great way of finding cell bodies, as well as getting a measure of like which brain regions are innervated or giving input. And the first paper that we published using this technique is a paper out of Rui Costa's lab, looking at a specific population of cells in the basal ganglia. This sort of signal to slow down in unique or sort of in novel environments. And so you can see we can get these really nice reconstructions of the basal ganglia and sort of map these cells using Imaris. And we could also do this in the looking at projections. So this is some data from Alice sort of gave me some data early on to develop this pipeline, and here we can actually map this sort of in the brain, and also sort of start to look at which brain regions these neurons are innervating. And then we can also sort of do some initial individual looking at slice by slice projections, as well as also doing sort of heat map analysis. I'll just go back there. And all of these sort of outputs, they're generated automatically. So this is a lot of this stuff just, the really sort of the best part about this pipeline in terms of ease of use is there's really only two steps. This sort of first step of reviewing the montage of the sections, and then a second step where you're basically doing this registration. And more recently, Anders has a paper that's been accepted in Nature Neuroscience using these projection analysis tools. So he was able to look at both CSN projections and spinal inputs and map which brain regions these cells are innervating. And we have a couple of other papers coming out using these same sort of techniques. And I guess sort of importantly too, like at the time, like machine learning was a good way of dealing with this problem, but more recently, deep learning has become more accessible, and this really has opened up two important avenues for people using these kind of tools. One is, we used to have to rely on a very strong label to, like DAPI or NeuroTrace or NISL to identify the tissue sections, but we've trained neural networks to detect the tissue in more difficult conditions, like just autofluorescence, or where there's just a few cells labeled, and that opens the door to being able to detect tissue more reliably. But I think more importantly, we can also use these deep learning-based segmentation tools to detect the cells and axons and dendrites, and so that makes for a better and more reliable detection of those processes. But also, for people who are doing studies like looking at CFOS, where detecting those cells, especially in very dense environments, is very difficult, using deep learning can give a much better result. And so we have developed like trained some networks for doing this, and BrainJ hasn't been fully adapted for the cell analysis coming from the slide scanner using neural networks, but it's something that is possible. And if someone's sort of eager to start using that, then that's something that I can add in. But just keep that in mind. We have these tools available, and I think it really makes projects a lot easier. And then finally, just a bit of a plug for a new pipeline we developed, sort of building on this. So we have these, even though there's some discussion about how good the atlases are in the mouse brain, analyzing data inside a common coordinate framework is just a far superior way than just analyzing images on their own and not being able to relate animals to each other or groups to each other. And we wanted to be able to do this in the spinal cord as well. And so I worked with Felix Feiderling and Carol Mason and Jane Dodd's lab to bring across a lot of the work I'd done in the mouse brain to be able to analyze the spinal cord. And so we could take some initial, 2D atlas work that the Allen Institute had done for the spinal cord, and we were able to digitize this into a full 3D spinal cord. And then we were able to use a lot of the tools developed in the BrainJ pipeline and some new tools that Felix added to deal with just some of the unique aspects of the spinal cord rotation. So we have images like this coming from the slide scanner. They can look quite messy, but after we put them through the pipeline, we have something that's quite a cohesive volume. And then this is something we can then map to the atlas. And this is something that allows us to, map cells as well as projections. And here in the slide, we're looking at, these are FOXP1 positive cells that should be localized in the limb segments. And we're able to sort of plot these from multiple animals and really show that the pipeline is able to work really well from animal to animal. So, obviously it's not a useful tool if there's a lot of variance, and this sort of just shows the robustness of the pipeline, and, really that we're able to map cells basically down to the resolution of the atlas that we have. And so, if you start to work in spinal cord circuits, this is another really powerful tool that we have. And this just recently came out in Cell Reports Methods, and we have a STAR protocols paper coming out in the next couple of weeks. So with that, if you have any questions, I'm happy to answer them now, and then we can sort of dig into doing the tutorial.
I just wanted to ask, how easy is it to build such a reference atlas? Because I don't work with mice.
Oh, sure. You mean like in like a different animal system where there's no existing atlas? Yeah. Yeah. Exactly. So to do it properly, you need to have a good amount of data. So like in the case of the mouse, it's over 1,000 brains that were imaged using 2-photon microscopy, looking at the autofluorescence, and then they create kind of an average brain, which actually lets you sort of see at a higher resolution than what you're normally imaging at, and you can start to resolve like features in the brain. And then once you've got something like that, you need to kind of annotate that, and that can be like coarse annotations or getting down to very fine annotations. And in fact, like this will actually become more clear during the tutorial, because I'm going to show you two different atlases: the Allen Brain Atlas, but also the atlas that the Kim lab developed. It's a little bit nicer for some brain regions, and maybe that'll help you have an idea of what's involved. Yeah. Okay. So, all right, so what I'm going to do is I'm going to share a view of the workstation 2 at the Zuckerman Institute, where we have BrainJ installed. And yeah, so basically like BrainJ, you can install this on, it really needs to be run on a Windows computer because of some of the tools we use along with it. So Ilastik doesn't work on a Mac, and Ilastik works on a Mac, but works in a different way. And so this has really been designed for a PC, but apart from that, you don't need anything special. To get the latest version, you can just go to GitHub and download the pipeline. And so this is the jar file that you'll need here to install it. And I encourage you, like if you're going to use this, hopefully after the tutorial today, you won't need much help. But if you do need some extra help going through it, then the PDF guide is just here as well, and this goes through a lot of the basic troubleshooting steps. And of course, you can always reach out to me if you're stuck on something, and I'm happy to help solving this. So the examples I wanted to share with you, just in case you didn't know how to install a plugin for ImageJ, so you would just install Fiji, which is a version of ImageJ, for those of you who haven't used it before. And so I can just open this up here. If you ever need to find like the plugins folder, like if you're not sure where it's installed, you can just go to the folder where this is installed. So the plugins, there's a folder inside Fiji called plugins. Hang on, this internet's been very slow. And then in here is where you can install plugins. So actually, for a lot of plugins that are like a bit different to the main Fiji list of plugins, I just put them in a folder here so that I can just copy them in. So here I've got BrainJ installed here. So that way it's easier to find them rather than looking through this big list. They'll still appear the same inside Fiji. Okay. So the first thing I wanted to show you is just a very basic example so you can have an idea of like how it processes the raw data. So in here I have a folder. So this is just 5 images from the slide scanner from a brain data set. And probably the first trap that people fall into, like if just getting, so you can use data from the slide scanner, it can be data from a confocal as well. Any data that, as long as it like, if it's from the slide scanner, it doesn't have to be like manually ordered. If it's from a confocal or something where we don't know this coordinate system on the stage, then you should sort of alphanumerically label those images. So if it's from the spinning disk or a point scanner, usually you're marking out those tissue sections anyway. So just make sure that you mark them in an anterior to posterior order, and then you'll be fine. For the slide scanner, because it's such a regular system and we don't have control over which sections it images in any particular order, we just read in the coordinates from the files, and that way we know where they were on the slide. And so if you're using the slide scanner, the ordering will happen automatically. And one of the first things to look for is just look at the files in this folder and see what their size is. Anything that's smaller than 100 megabytes, I'd be a little bit suspicious. That could be like some debris or maybe the corner of a slide. So I would just look at the folder and just sort them by file size. Look for the small images. Just check that they really are sections if you're not sure. If you run the pipeline, it'll probably.
Yeah, I think it's pretty good.
It'll show you anyway.
Especially the end, and the beginning looks bad, but there's no whiskers in the frame, so you're not going to see them.
Yeah, I was going to say it's just because it's not really there.
Actually, here the other side is there a little bit, but
it's not really
we weren't focusing on that. But these, especially a little later when they really come into the frame,
yeah.
I'm just looking for to mute
other thing to keep in
Does
mind.
Anyone know where you mute other people?
We don't. We're thinking about 2 lights or 2 cameras or whatever to like focus on each one.
You don't see the option now, but normally it's in the
participants. This is a much more clever way in that you don't actually have to process the video in a way where this and this is optimal.
Okay, there we go. Sorry about that. So, so just sort them by order, have a look for the small ones, and then also look for the large ones. Like if there's anything over a gigabyte, it could be likely to be 2 sections or 3 sections. And if that's the case, that's easier to solve by, talk with us about the way you're scanning those sections, because we should be able to get it to a point where you don't have double sections during your scans. The first step in using BrainJ, so if once it's installed, it'll be in plugins. And if we scroll all the way down, actually it's a bit hard for me to see here. So it'll be down the bottom here. There'll be an option here called BrainJ. And the very first step here is to set some parameters. And this is important because the way we really wrote this is so that you can process multiple brains at a time. So the idea is you could have different brains from different experiments that were captured in different ways. And you can just, run this step quickly for each brain and then just select those folders and run it overnight. And really you're just answering a couple of basic questions about the data set. In this case, we're going to run this data set here. So I can just select the folder. And we're doing whole brain analysis, and coronal sections is really the best way to go. Sagittal sections we used to do, but it's always very difficult to get the alignment nice. So I really recommend using coronal sections. The 2D slices, and then we need to figure out if they need rotation or any flipping. We need to know the cut thickness. And if you're doing something where you're using every second tissue section, so maybe they're cut at 50 and then you're using every second one, then in that case, you would make sure that you would say 100 here. In this case, these are kind of just random sections, so it's not so critical, but definitely this is really important when you're processing an actual brain to have the correct value in here, because that's going to affect the alignment to the atlas. And we can also do some resampling. A lot of the cell analysis and the projection analysis works fine at the 2 microns per pixel. So this just really helps speed up the pipeline. But you can use high resolution as well. It's just going to take longer. And we also need to tell it which channel is the channel that has the DAPI or the Nissl or whatever is being used to sort of show the tissue. And what the intensity of the background is. And actually, I'll quickly show you how to look for this. If we open an image up, sometimes these larger 2-dimensional images can take a few seconds to open up in Fiji, so you just have to be patient. The way that you can check for the background, so on the slide scanner, usually the last channel will be the DAPI channel. I'm just going to adjust it so that I can see something. So you can see there is some background here. We can do this automatically, but it works out to be a lot better if you just manually look for this, because it'll make it a cleaner job of segmenting the tissue. So this background for this sample, in case you don't know, in ImageJ, you can see the intensity value just here in the status bar. So as I'm hovering the cursor, you can see that this is hovering around 200 or so, maybe a little bit higher. And when I go into the brain, it immediately jumps up to 500 and then up into the thousands. So it'll be, pretty safe to put in something like 230 or something like that, and we should get rid of most of the background. And we also, you can see we need to rotate this to the right. So that's the 2 sort of things I need to know there. So I'm just going to go back. I'm going to go into BrainJ. Okay, so I'm just going to choose the directory. This is all fine. We need to rotate to the right. We don't need to flip. This is fine. Our counter stain channel is 3, and the background channel, we can just say 230 here. So that's it. If I had other brains, then I would just do this step again. And just so you know, if you need to change these, the easiest way is just to right click and edit this. These are just a simple like CSV file. So if something doesn't work here, maybe the background wasn't very good, you can just come in here and change the value here and then save this file, and it'll be an easier way of reprocessing the data set. And so now I'm just going to run this first part. And here, this, if you had multiple brains, you would just, select multiple folders here and then run it. And what you'll get, so straight away, like you'll get some information on the version that you're running. So that, this is a file that's saved in the folder. So 6 months down the track, if you need to go back to these data sets and figure out what was going on, or maybe it's a year later when you're working on the results for the paper, this information's here, and it's easy to sort of track back to what you're doing. And it'll tell you a little bit about what's going on. So it's important to sort of know how long these things are going to take for you to plan your time. And so I've tried to make this, as clear as possible. So, here you can start to see how long it's going to take just for running this. But also at the end, it'll tell you how long it, took to run the brain, and that way you can start to think, okay, if I've got 3 brains, it's going to take till tomorrow morning to deal with this, or just so you have a little bit of ability to plan around what's going on here. And this is kind of a typical time. So it should be, for doing this reprocessing, 20 seconds is pretty typical for a 3 to 4 channel image from the slide scanner or from the spinning disk. Does anyone have any questions so far?
Hey, Luke, I have a question actually. Now I'm thinking that you were running it. So let's say that we know that it's working nicely. I don't know, we test probably on few slides. So we want to run it on the entire, and a big folder with a lot of images. Can you run it on the background? Can I just set it up in a way that runs on the background, or needs to run, I need to leave the computer basically working on it?
Yeah, I mean, you can run this in the background, and you can, I mean, I know some people run.
I guess my question is like how demanding is in terms of memory.
Oh, yeah. I mean, we should have put this. I don't think this step shouldn't be too bad. I mean, this is a pretty powerful. I mean, CPU says very high here, but I think it's about 25% of what this computer can do. I know some people run two or three of this step in parallel. Like you can just open up multiple Fijis and run it. For this step, it's fine. For the other step, there's some steps where, for the other part of the pipeline, the sort of final step, some of those processes can be very intensive. And so I would just leave that to run without doing anything, like use a workstation that you're not using to do other things. You're referring
to the part of the machine learning, maybe?
Yeah, the machine, the sort of alignment to the Allen atlas, that only takes like 5 minutes, but it's quite heavy. And then, yeah, some of the other projection mapping stuff. I mean, I run it while I'm working on documents and things like that. So if you're you know, writing a paper or something like that, it's okay.
Okay, thank you.
Yeah, and actually so when it's finished, it'll tell you that, you know, so it makes a preview, which I'll show you in a second, we can use for troubleshooting. And it also prints out the sort of dimensions of all the images, because sometimes, you know, because these aren't listed in anterior to posterior order in the folder, it can be really hard to track down which raw data file is causing the problem. So if you have some unusual data that's causing the pipeline to have trouble, this list here, you can look through it and say, okay, you know, if one of these says 20,000 instead of 10,000, we know that's way too big for a brain section. So I would look at that image and see what's going on. I can just see what the file name is and have a look. But this one, there's nothing that's out of the ordinary. You know, if you're using the new pipelines from the slide scanner, where we also give an image of the pre-scan as well as the raw tissue sections, you want to take that pre-scan image out, because that's going to be confusing for the pipeline to have pre-scan images as well as the individual sections. And I think that I've written the pipeline now, so it actually will give an error if there's data like that. It'll just stop and tell you that there's something like that happening. Okay, so we've run this first part. The folder we want to go into now is this section preview folder. So this contains the data we're going to be analyzing. This is kind of a preview to help you deal with the big data sets. And there's a montage and a stack view. So if I go into the montage view here, this is just the 5 sections. So now we can, you know, it's adjusted a little bit for us, but I'm just going to adjust the channels so I can see them a little bit more clearly. And so this isn't a high resolution image. It's really just a sort of macro view, so you can quickly assess what's happening if the sections look good. You know, in this case, all the sections are fine, but I'll explain to you what to do if things aren't fine. You know, so this is a place where you can check, okay, which sections need to be flipped. You know, maybe section 4, there's some artifact like a bubble or some big bright blob that is going to affect the alignment to the atlas. And I'll show you how to deal with those problems. In fact, like in the manual that I put together, this is actually covered. So yeah, so just here, you know, actually the pipeline's pretty robust to this first issue. But this second issue here, if there's tissue that's really touching the main section, what's going to happen is the sections are going to be registered, but then when it's been aligned to the template brain in 3D space, this is going to look like it's part of the sort of outer cortical layers of the brain. And so the atlas might try and map onto this, or it probably won't map onto it fully, but you're going to have some deformation here, and that's going to affect the accuracy in this local area. And so if you want to avoid that, then we would just manually edit that channel. And I'll show you how to do that now. You know, the important thing is you don't have to do this to every channel. The channel that's being used for this alignment is the DAPI channel in our case, or channel 3. So if I saw that, you know, section 4 had some kind of artifact, I can just open this image up, adjust the brightness so that I could see. You know, maybe we could treat this as something that shouldn't really be there. I can just like draw across this region, and then just press Control X to cut it, and then just save the image. That's all you have to do to correct any of those sort of artifacts or minor things like that. And if you're placing your sections like well spaced on the slides before you scan them, then this should be a fairly rare kind of problem to deal with. The second one, which is probably more common, is if you've floated these sections and you're not sure of their orientation. That's something where you'll need to flip the images later. And that's something that we can deal with in a bit more automated way. So obviously you'd look at that montage image. I'm just going to close this. Go into BrainJ, and then here we've got this tool, this sort of section flipper and replacer. So if I open this, it remembers the brain that we were just working on, and we can just tell it here which sections need flipping. If anything happened and some of them are vertically flipped, you could fix that here as well. And then, if you've got big data sets and sometimes things go wrong, a section gets badly damaged, and you still need to preserve the whole data set, you can replace those with neighboring sections here. And you also have an option here of how you want to do that. Do you just want to replace the DAPI or the NISL, but clear the other channels so that you don't have duplication of the cells? Or do you want to just duplicate those cells as well so that it's sort of consistent through that area of the brain? And so that's something that you can choose here. And if I run this, this is a pretty quick process. And it's just repopulating the images here now. And then it's going to create an updated montage view. So we can actually look in here now and see the updated montage. And then now there's going to be one that's upside down here. This one's been flipped. So this is how you could fix that problem if you had those problems. And that should be a very quick process. And then finally, we want to do the registration and the analysis. And so we need to run a new step now. So the idea is you would do this step for multiple brains to get to this stage, and then you would go through and set the analysis settings for the brains. And this window has a few options on it. And I can just walk you through these. And we'll go into these in a little bit more detail as we sort of focus in on each of these steps later. But obviously, tell it the folder that the brain data is in. We need to tell it a reference section. So the first part of these pipelines is to really register the sections all together in 3D space. And so we need to tell it a section to start. So it's nice to sort of pick something that's in the middle of the series so it can work forwards and then work backwards. And just so you know what I mean, like if I was registering this data set, actually, I'll even do this properly. I'll just fix this. Oh no, I'll just leave it be and I'll run the registration. So if I would pick something in the middle for this case, so if I set the analysis here, so we have 5, so I'm just going to say 3 in this case, but if you had 60, maybe somewhere around 30 would be good. This background subtraction, I'm going to talk about this a little bit more in a second, but this helps us to detect the cells and the axons and the dendrites better than if we didn't do some background removal. this is if you want to generate a full resolution image of the DAPI channel. So for a lot of people, they're just interested in counting cells or measuring projections. You don't need to have the DAPI channel at high resolution, but if you were planning to do something with it, or maybe it was new-n and you actually wanted to count cells on that channel, then you would ask it to actually create that at full resolution. Otherwise, we can disable that and save some time. And yet we have another option here to do a second pass registration. So sometimes just one pass of the registration, if the data is quite messy, it can help to do a second pass. This is slower, but it has helped in some difficult edge cases. Then we have an option here for the cell analysis, and we offer a couple of different things. So we can do a fine maxima, and I'll show you kind of how that works. This is very similar to kind of how ClearMap would work, where you basically have a fluorescent area or a volume, and you're just finding the brightest sort of point inside that volume. And then using machine learning, which is definitely the best way if you've got like these virally labeled cells and you're trying to differentiate between cells and axons and dendrites. And we also we have like a manual cell count here, so you can do all this sort of registration and then just manually click on maybe where you've inserted a probe or just a small number of cells if you want to be very careful about it. So these are the sort of options that we have. Actually, I might go into this in a little bit more when I go through the Ilastik and cell segmentation. But basically, these are the channels we want to do the cell analysis on, and there's some settings here which I'll describe in a minute. And same for the projections. We can do a sort of binary threshold where we really just say everything above this intensity level, that's what we're going to consider an axon or dendrite. Which could be okay, but that leads to like a bit of over segmentation. Or we can use some machine learning where we can be a little bit more accurate about which pixels are indeed the axons and dendrites. And then we really just need, in this case, the threshold here for machine learning would be, rather than an intensity, it would be the probability that that pixel is an axon or dendrite or not. And this will be like more clear once we look at Ilastik. And then finally, and this, you shouldn't really need to change these at all once you've set them. This is really the location of where Ilastik is installed, and then the location of Elastiks, which is what does the registration part. And if I run this now, then it will have created another file in here. So we should have two files in here. One that's sort of our basic parameters for the data set, and one that's the sort of settings for the analysis. If I close this. So now I can go back in here. And here, so this is the final part of the pipeline, and this is something you would again run in batch, and you would put multiple brains in here that you were processing. And for a full brain, typical type of, analyzing all of this sort of stuff, it should be about, an hour to an hour and a half of processing time. Depending on, if you section very thin sections, then it could be more than that. But if you're doing like 50 to 70, then that's kind of a typical time. We can put the data sets in here, and then we can choose which atlas. And I'm going to show you, like, we can use both the Allen Brain atlas, but also this enhanced and unified atlas that the Kim lab made, which is actually a little bit nicer if you're interested in subregions of the striatum and actually some of the different motor cortical areas and sensory areas. And I'll explain that momentarily. So this is kind of set up so that you can do individual things or just do everything. It'll also, if you've already done this on a brain, it's going to see that you've done it and just move forward anyway. So you don't have to uncheck these if you've already done these steps. It'll figure that out. But in our case, if we just wanted to do just the section registration, I can uncheck these. And then just run the pipeline. And again, it should explain to us here, I've made this so that the registration process should be very quick, but translating the full resolution data can take some time. So, this is just 5 sections, so it's only going to be, it's less than a minute per channel, but it'll give you an indication here of how many minutes it's going to take for the full brain. And maybe that'll be like, depends on how many sections you have, but this could be 20 minutes or so per channel if you've got a big data set. And this should be creating a folder here called registered sections. If the way that this has been written is so that it's not a black box, so all the data is left here. And then once you're sort of happy with how things are looking, if you've confirmed that things are working well, then you can delete some of this data. So, once you're confident that things are well registered, you don't really need to keep this reformatted sections folder. And once you've sort of done the processing, like the cell segmentation and things like that, you don't need to keep this registered sections either necessarily. Okay, so we've processed this now. This isn't necessarily going to be so nice because some of those sections were upside down, but if we look at this series, they should be, at least, if there were sort of serial sections, they would be nicely registered, and I'll show you that in a second. But also, we all crop this right around the sort of minimum area of the tissue, and this is also important for getting a good registration result, as well as, just keeping the file size smaller later on. Okay, so we've gotten that far. Now, if we wanted to actually analyze the cells, let me show you. I have to try and find a good area just to explain this. I'm just going to duplicate this region here. So I'm just pressing Control-Shift-D to duplicate this region. If we were to try and use something like so this is the raw data, and it's important to realize we actually do a background subtraction before we do any analysis. And if you want to sort of see how that's working, I mean, this is background is a really big problem in analyzing images that come from biological samples. And, you can see a really good example here, like there's some faint cells out here in a dark background, but then there's some faint cells here in a bright background. And if we try and set a threshold that captures some of these faint cells, we're going to end up detecting this whole thing as one blob. And you can easily visualize that. If I try and threshold this now, like, this is just an automatic threshold, but it's you can see, like, if I try to get those cells, it's going to be a major problem. And one way to deal with this is to do a background subtraction. And let me just find a good way to, so I'm just going to duplicate this again. Just make this a little bit brighter so it's easy to see. And then I'm going to use something called background subtraction. So this is a rolling ball filter, and for the images of this size, we usually use a ball radius of 7. And you can kind of see visually what it's done here. It's gotten rid of a lot of the low frequency information in the image. So if a good way to sort of visualize this, if I just draw a line through these cells, try and get a more faint one. You can see like this kind of low frequency background that's increasing, but you can see the sort of peaks of the cells in this. If I was to look at that on the other image, yeah, so you can add these just by pressing T, you can add a region to a list. And then when you click on another image, you can add this in on another image. So that's a nice way of copying regions. And I can now sort of plot that again. You see really well here what it's done. So it's kind of like, the best way to describe it is it's kind of like rolling a ball across the image, and you're setting the size of that ball. And if you kind of imagine, sort of, if you flip these, you can imagine the ball rolling across this surface. And we've made it big enough so that it doesn't fall into these valleys. And that way it can sort of record what the slope of this sort of background is, and we can subtract that away. And we're left with then peaks that are sitting nicely above 0, and it becomes a lot easier to do the analysis. And one of the most basic ways of then counting these cells, sorry, the mouse has a little bit of lag, so sometimes it's a little bit funny to use here. Okay, so the way that you can, if you want to count the cells very quickly, say you have a really easy sample and the cells are just really bright, they're very sparse, you don't want to use Ilastik, then the one option we have is to use like a maxima detection. And you can see this very easily. If I just run the maxima detection from image J here. And we need to like obviously set some kind of so you see it's not doing a great job here. It's picking up cells along the projections, but you can kind of get the idea of how a maxima detection would work. It's finding these local regions of bright signal. This actually works well when the samples are very easy, but for most of you, you won't have easy samples. And so this is when I would recommend to use something like Ilastik. And so this is where I can actually show you how to use Ilastik. Actually, maybe we can even, actually, I'll show you how to generate the data. So, if you're going to use Ilastik for training for cell segmentation and axon dendrite segmentation, you don't want to use these files as they are, because in the pipeline we're applying that background subtraction. So what you could do is like open these images up, do the background subtraction, and then save them somewhere. But because this is a really common thing, there's actually a step in BrainJ to save you that trouble. So if you go in here under other tools, there's an option here, create Ilastik training images. And then you can just say, okay, this is my my data set, and we can just say, all right, I want channel, it should be just like this, 2 and 3. And now it's just going to process these ones here. It should make a folder out here, Ilastik training images, and then process these for us so that we don't have to do that. Let's just see. There was some changes to it. Oh no, it's working. Yeah, if you do run into any problems on the pipeline, don't hesitate to reach out. Every now and then, ImageJ is constantly updated, and every now and then there'll be an update that breaks part of the pipeline, and that's usually just a simple fix that I can take care of. So, if you do hit a wall using it, don't be afraid to just like send me an email, and usually it's a simple problem that we can fix. Yeah, these are big files, so like running the background subtraction is not necessarily quick, and so this is a good way of doing it without having to spend like 20 minutes at the computer opening and closing images and running the background subtraction. So I've tried to make this as like painless as possible. Yeah, so it's working away now, but I can open up one of these images.
Have something in the meantime?
Yeah, sure.
So did it now use the radius of 7 that you previously set?
Yeah, it'll look in the folder and see, okay, what's this pipeline for this brain going to be doing? And if I open this up, it'll see, okay, background subtraction is wherever it is in here is set to 7. So that's what I'm going to use to make these files. And it's just as simple as that. It's just that one step really gives Ilastik an advantage in being able to do the pixel probabilities. So it's definitely worth doing. Okay, so what I'm going to do now is just show you how to use Ilastik for this. So it'll automatically create a folder called Ilastik projects, and on the resources on GitHub, you'll actually find a link where you can download some working Ilastik project files. These need to be copied into the folder because as the pipeline's running, it will look in this folder here and find the project for the channel that you're analyzing. And in fact, I can actually look in this. I've got some already set up here. So here's some Ilastik project files, and the naming convention, like it's in the README file. I think it's this one here. So for channel 1, it would need to be named channel 1, and for channel 2, it would need to be named channel 2. You know, I'll walk you through that. Like, it'll be fairly quick going through Ilastik, so you know how to use it. But if you want to know in more depth, then there'll be a specific tutorial for this. But the Ilastik website actually has really good documentation, so I think they've tried to make it as easy to use as possible. You can just download it. There's like an Ilastik, you know, they've got a website where you can download it, and they have some videos and steps that really take you through this process. I'm just going to run it now. Sometimes, like when you open up a project, it won't open the project. So, you know, this is good for you to see. Like it can be a little bit glitchy sometimes. So, you know, it's good for you to see how it's working and how to sort of deal with these problems so you don't get put off using it, because it really is a fantastic tool. Okay, so even though I opened that project up, it didn't run. I wouldn't recommend, so this is what we're doing. We're doing pixel classification, which is where we train it to classify pixels as either like, you know, foreground or background or cells or dendrites or whatever we classify them as. I wouldn't start from scratch for BrainJ because there's a few things I've changed in the project to make sure it works really well. So I would download the example project, clear the data out, and then put your own data in. And I'll show you how that would work. So I'm just going to open the project. Okay. I'll start from the beginning and just quickly go through here. Basically, this top section here is where you can copy the data. So, you know, in our case, where we've just made these images, and I would recommend if you're just working on one brain, I would use like 3 example images. If you're working on, you know, a series of brains, I would take sections from different animals in the group just so you can train across, across the group. Because you want this to be like as robust as possible, and you don't want to be changing it for different animals. You want to be, you know, doing this training and then running it as it is across all the animals. So in the training images here, I would just go in here and grab an image and drag it into the project. Really important step. So if this was starting from scratch, I would like select the old data, and just hit reset. This will just clear these images out of the project, but leave all the settings intact. And then when you add new data in, like this one here, you can see it's just linked. It's not actually copied into the project. And this is actually a big problem. If you forget to copy the file into the project, you store this project, you delete the image or you move the image, this project then actually becomes difficult to open and use. So, you know, especially if you spend some time training, you want to be able to keep that work. So I'd recommend just saving this image into the project, which you can do by clicking Edit Properties and change the storage to Copy to Project File. And if you do this, then you can save the project, and this image is now embedded in the project file. And that means you can take this project anywhere, and you can let you validate how the training has been done. It lets you modify it or improve it without having any errors. So this is the first step. The second step is to set some features. So one of the powerful things so this is a random forest machine learning tool. So it's not deep learning or anything like that, but one of the sort of powers of this is it uses, in image sort of segmentation, we usually apply a filter that makes the image easier to sort of binarize or segment. What Ilastik does is actually apply many different filters. So some of the more typical filters like smoothing and median filters or things like that, but it also uses filters that help to extract structural information from the image. So they're not truly like texture filters, but it does help to sort of identify different textures on the cells. So maybe the soma has a different kind of pattern to it than another cell or the dendrite and things like that. So it becomes a really powerful way of filtering the data, and then you train it by basically telling it, okay, this pixel is a cell body, this pixel is an axon or a dendrite, and it can figure out what's different about the intensity values across all of those filters to help sort of build up an idea of like, okay, this pixel has these intensities on these filters, so it most likely is a cell or an axon. And if you wanted to, you can visualize these filters here. If I click here, you can see what some of these filters look like. And essentially, like, we use this is like just to make it work quickly and as well as doing a good job, but you can play around with these. If you're not happy with the result, you can include more of these or less. And then we can go into the training. So the way that this works, actually, let me just get rid of these. So we have, the way we've set this up, we have the cells, the projections, and background, as I sort of mentioned already. It can be a little bit hard, like straight away, it can be hard to find the image. So a good thing to do is to come right down to raw input, right click, adjust thresholds, turn off automatic, and start manually adjusting this so you can actually see what's in the image here. Hopefully, yeah, so now we actually can see where we have some labeling. The controls for this are a little bit funny, but it's shift left click to move around, and control and mouse wheel to sort of zoom in and out. And actually the sort of easy, like in this case, less is more. Like you really want to just train a few pixels and assess what's happening and then start to sort of refine that training. And so I can show you very quickly, like if I just train a few cells here, this maybe is a little bit more saturated than I would normally use, but I can see like where cells are. And then I can see, like, I'm just clicking on projections, just clicking a little bit to sort of help figure out what I think a projection is. And then obviously background. And I would be careful about where I place this. I would be like trying not to put it anywhere where there might be some projection. And this is useful to get rid of things like bubbles as well as like the edge of sections and things like that. And now I'll just click live update to see like how it's actually performing. And you can see like, it's doing okay. It's not fantastic, but it's going in the right direction. So now I can then start to like say, okay, actually this is a cell. It's not really a projection. I can see areas where it's sort of on the fence, and that's where I'd focus my attention. And then I can start to try and refine this a bit more. You can see some areas here where it's like really, if you make a mistake, then try and erase it, because if you have got a pixel somewhere that really is in the wrong place, you're going to be fighting with that the whole time. So even just one pixel in the wrong place can cause you some trouble. And then to tighten up things like around the cell edges, I would really just tell it, okay, like right here, this stuff is really background, and then you can really start to tighten up where the cells are located. So I won't sort of spend too much time on this, but you get the general idea of how this can work. And this works, reasonably well. The areas where like you might, so we can get it definitely better than this, but the areas where you might still have trouble is very dense. So maybe areas like this, they can look, this might not be a cell in terms of what it's obviously an axon or dendrite, but if it's really sort of cell shaped and it's the same sort of intensity and the same pattern, then it's going to look like a cell to the pipeline. And the only sort of improvement on this is to really start to use deep learning at the slide scanner or the confocal. So that's something we can do, and that's kind of where I think that if you need to improve it, that's where it can happen. But we can get it so that all these are picked up as projections. Okay, so that's it. We've trained it. I would save the project, and then I would run the sort of latter part of the pipeline. And I can sort of show you now. So then, we would set the, okay, actually, let me show you. So if we were going to do the cell and projection analysis, we would need to say, okay, these two channels, I want to do cell analysis. These two channels, I want to do projection analysis. The threshold here, if you're using machine learning, is the intensity, but that intensity is like the probability of it being a cell. And I'll show you how to check that. And same for here. This is a probability of the pixel being an axon or dendrite. And the other one you need to think about here, this is the cell area. I think generally 20 microns works well for cell bodies, and 15 works well for like nuclei. But you can play around with this if you're having trouble getting the results that you expect. So how to check this, if you don't know the answer to this, sort of like what the threshold should be on the first brain, then you should really, run the pipeline. You can just run the first step. So you're, doing the section registration, you're doing the atlas registration, and you're doing the cell detection. This is going to run Ilastik and create the Ilastik images. And if you look in, if you open up the data set after you've run that part of the pipeline, you're going to have a folder here called process sections. And in here, this is kind of data that you can delete after you've run the pipeline, but it's really useful for troubleshooting, like what's happening and how and refining the pipeline. This folder here, probability masks, it actually contains the images that have come out of Ilastik. So if I open one of these up, and actually I'll go a little bit further in. So you can see if I zoom in here, what these images look like. And if I split this into separate channels, this is our background, this is our like projection channel, and this is our cell channel. What you can do is determine what that threshold should be. So if I like zoom in here, I can hover the cursor and actually see like, okay, this is where it's very certain that these pixels here are cell, and you can see the intensity is around 200 or a little bit higher. Out here, this is where it's not really certain, and you can see they're around 35 or 40. So, this one's a little bit lighter, maybe 190. So you would look at this image and go, okay, maybe it needs to be 150. That's where I draw the line, and that's where, I'm going to be considering those as cells. And same for here, like if I look in here, we should have kind of like donuts where like that's where the cell bodies will be, where it's more certain that that's a cell, and then areas where it's more certain that these are, actually this image is actually out of focus, that's why these are kind of blurrier, but you can see where it's actually more certain that these are projections. You would set those, and then, you would basically, once you're happy with those thresholds, you can just run, like select all of these and just run the pipeline across the multiple brains. And like I said, this should take about an hour to 2 hours per brain. And actually, let me just show you. So inside, so this is an example data set that's already been run, and you can see in here, so it'll save a log file so that you can actually, as it's running, it will tell you what it's doing, but also you can look at the log file later to see, how it's running, what it's doing, how long it should take. At the end, it'll tell you how long it's taken. If there's any errors, it's going to tell you, one thing to look out for is like, was the alignment good or not? And I'm going to show you how to check that now. It tries to report as many things as it can to help you troubleshoot things along the way, because these data sets are quite complex and there's many things that can go wrong. So just trying to make sure that it's like easy to use even with difficult data, it's been quite important. Okay, so let's say we've run the pipeline. This is what we get, this analysis output folder. And these are the outputs are sort of set up into subfolders so that you can easily find the data. The first thing that I would check is this image here, template brain aligned. And this is really useful because it's going to show you how well the alignment has gone. So in red is our DAPI channel in this case, and green is the Allen Brain Atlas, you're always going to have a little bit of a misalignment on the first image, just like these are like the beginning and the end, where, you know, if there's, especially if there's tissue damage, it's going to have some trouble. But you should find that like through the main part of the data set, the alignment is good. If there is a problem here, or if the registration fails, I would go back and check, what does my registered data look like? Just looking, you know, the best one to look at is like this, the DAPI folder. Just like have a look at this data set, you know, and go through it and see like, okay, how does it look? Is there sections that are like completely damaged in the middle of the series? That's going to cause a problem for the registration. And if that's a case, I would replace that section, or, you know, right at the end where, you know, if it can be difficult to section and keep some of the tissue intact towards the posterior part of the brain. If you're having trouble there, you might have to just truncate the data set and delete those sections so that you can get good alignment for the main part of the brain. So that's the first thing that I would check. If that's good, then you can kind of trust the alignment has worked. So the cell locations and things like that should be good. And so for cells, you get a bunch of outputs. Like, you know, obviously, if you're just interested in plotting the data, you'll have a CSV file here that has every single cell, its location. We also measure the mean intensity across all the channels. So if you're looking, say, like populations of cells where there should be some co-expression or co-labeling, then you can actually use these intensity outputs to say, okay, like, you know, I'm interested in cells in channel 1 that also have a high intensity of channel 2. That's my double labeled population. You can look at that in this, you know, these intensity columns. We do give like the bregma location. You know, this is just based on comparing those atlases. So, you know, this is, you know, as accurate as we can get it, but just be aware that this is like a calculation to get these. And we also give like the hemisphere and the ID, and then the acronym of that brain region. And then we also give like a summary. So, you know, this is total number of cells, the brain region, the acronym, like all the sort of information that you'd be looking for. And just remember, like, even if you just want to have a quick look at these in ImageJ, you can always sort, you know, these tables. So you can say, okay, which brain region has the most number of cells in the right hemisphere? You know, it always sorts ascending, so you can have a look on this side. So there's actually not many on this in this hemisphere, but you can see the regions here. And then we also plot these. So there's like some basic Z-stacks overlaid with the template, or you can just quickly look at the 3D brain and just see like, okay, where are these cells? These are just like quick renderings so that you have something, you know, quick to check how the data looks. But of course you can render these in other programs as well. And then we create the heat map as well, which is useful if you're trying to like get an idea of like cell density. You know, sometimes these images will be black when you open them. That's because the intensities might be a little bit low. So you may just have to auto scale. So these can be used to do kind of like group comparisons of cell density and things like that. For the projection analysis, same kind of thing. We have these tables that measure the volume of the brain region and the projection volume. And actually, it's important to note this is like a reverse mapping approach. So we actually, rather than sort of transforming the data into the atlas and measuring it there, that the raw data in that case would be kind of deformed. I'll show you what this means in a second. We actually bring the annotations back into the raw data and measure the brain regions and the volumes in that data set. And that way we can get a true measure of the volume of the region as well as a true measure of the amount of projections. So that's what's reported here. And then what's kind of nice is also to have an idea of like the density and the relative density. So the relative density is how much of like that total volume of projections is in the brain regions that it's sort of located in. So, you know, if most of those neurons are located in one particular brain region, then the relative density will be high for that particular brain region. So this is in a sort of percent. And then of course we have like all the acronyms and everything here. So this is easy to like put into MATLAB or Python or whatever you use for plotting. And there's also some things in here for validation. So this is kind of an overlay of the annotations with the projection. So you can look at this and quickly see, okay, like, does this make sense? Like what's actually happening with my data? And you can see here, like, okay, I can see that where these projections are visually. It gives you some confidence that things are working well. And we also have like a heat map. This is kind of just like a region by region heat map, but it's kind of useful to just sort of show, you know, which brain regions have got a high density of projections. Does anyone have any questions so far? It's all clear? Okay, so because like it's sometimes nice to look at this data in the common coordinate framework in terms of like the actual atlas space, these are sort of some renderings just to do that. So this is where we transform the raw data into the atlas space, and this lets you look at the projections in a sort of 3D space. And, you know, for some projects, we've taken these images and put them into Imaris to make some fancy sort of renderings. But, you know, you can just very quickly have a look in here and get an idea of, okay, like what's happening with this data? Where is it? But the real measurements that you'd want to be using is inside this density analysis. Likewise, you know, it's nice to look at the raw data in this way as well. So these images in the raw data folder are actually the raw data transformed into the common coordinate framework space. And I can just adjust this so you can see. So, you know, now we've sort of morphed this into the atlas space, and then you can just create a 3D rendering, or you could put this in Imaris as well. So now we have kind of like a volume, you know, looks like something that's come from the light sheet, but this is just reconstructed. Okay, so done the raw data. A region, you know, some people just like to, you know, look at the mean intensity of a brain region. Maybe that's also a good indication of how much cells or projections are there. And so this is all this is just measuring the intensity of those brain regions. So, you know, intensity left and right, and then, you know, an image of that, so you can quickly go through and go, okay, great, that's where I have the, you know, high intensity, and this should, you know, correlate with like where the cells and the dendrites are. Okay, so that's kind of the main sort of analysis outputs here. These, just so you know, these images here are kind of just the volumes of the brain region. So if you needed to know how big that brain region was in your particular animal, that's going to be contained in these files here. You know, one of the nice things about this is we can also use the enhanced atlas. So actually, I can just show you. So these atlases, we've sort of already been looking at this. These are kind of like this is the template file that we're aligning to. You know, if we go through this, this is kind of the average, you know, made by averaging many of the mouse brains using 2-photon microscopy. And then these are the annotations that have been, the most recent annotations that have been provided by the Allen Brain Institute. You can also analyze using the Kim atlas. So if you need to do that, it's just a case of, you know, in here running this step, but then just choosing the new atlas. So you can just choose, okay, I want to run the other atlas, and then you can just you know, compare results. If there's something that's sort of interesting to you about this atlas and you want to just, you know, cross-check with the other atlas, then you can run it with both. All you would have to do is in here, in fact, I've run it with both in this case. I would just name this folder as, you know, I would just rename it, and then rerun the pipeline with the different atlas, and then I would have another folder with the enhanced atlas output. This atlas, just so you know, it's worth looking at the paper for this. I can share a link after the tutorial. But it's nice for a couple of reasons. There's they subdivide the striatum, but they also there's some areas in the somatosensory cortex and motor cortex that are better defined in this atlas. And I'll just show you. I'll bring it up so you can see. So these atlases can be sometimes a little bit hard to look at. What's useful is if you just use a different lookup table, Glasby on dark. This is a useful one. Yeah, so one of the things, we often look at things that are in the striatum. So here, like this is actually a big region that hasn't been subdivided. But actually, if I pull it up to like some areas where we can see some of the differences, so you can see some of these divisions included here. And then there's some big differences in terms of like some of the cortical areas. So if there's areas you're looking at and you wanted to get a different perspective on where your cells are located, or just do some comparisons, it's worth having a look at this atlas. Yeah, so that's kind of like the general gist of what the pipeline does and how to use it. I didn't touch on this, but finally, if you were kind of doing some analysis where you wanted to extract one of the brain regions at high resolution to, compare with other brains or just make a high resolution figure, you can extract a specific region. And so this will just do the reverse mapping and pull out that brain region. So all you need to provide here is the ID number for that brain region, and then the pipeline will pull that out and save it in a folder for you at the full resolution. So that's something that is like a more specific thing, but that's here as well. Yeah, before I go on to the final like other tools, does anyone have any like questions about what I've shared? Okay. So like other things that people are obviously interested in doing is measuring volumes in the brain. Maybe you're interested in where a GRIN lens is placed or where a probe is being placed. And sure, you can like run the normal pipeline and you'll get like the raw data output. So you might just be tempted to say, okay, I can just look at this raw data and make my, like I showed you before, I don't know if I have it here, but just looking at sort of transformed data set. And that's fine if you just want to get an idea of roughly where it is in the brain. But if you want to make any measurements on volume, it's much better to be doing the reverse transformation and measuring the raw data. And this is not necessarily so trivial to do. But I've updated the pipeline to include this for doing volume measurements and like measurements of like, probes and things like that. So these extra tools, they're actually available just underneath other tools here. So there's a couple of things in here, like there's some stuff in here for doing like, if you need to do something from another data set, like light sheet data from Amaris or something like that, you can import data this way and using these tools. And if you just want to like, maybe you're imaging on the slide scanner and you don't want to use this pipeline, but you want to be able to just order the sections anterior to posterior and put them through something in MATLAB or Python. There's just a simple script here for just doing that reordering so that you can have the files in order and then run them through a different tool. And then there's also this volume measurement. So this is, like I was saying, for measuring things like, I'm going to use an example of a stroke, but also the GRIN lens placement. And so to do this, you would just pick the brain. So in my case, I've got an example here. It's like a data set that Alice shared with me containing some stroke data. And I would just select this folder and the channel that I want to analyze. So I'll just quickly show you this data set. So here's our data set here. And this is a data set where there's been a stroke induced in the animal. So you can see this volume here that we'd like to measure. And I guess you could just, threshold it, but then if you want to know about like where it is in the atlas and things like that, then you can just use BrainJ. So you would, use the initial steps to register the data like we have here. And then you would just need to do the, I can show you what steps we had to do. So we'd run the final step here. We would just do a section registration and an atlas registration. The atlas registration takes like 5 or 6 minutes, and then you're done. And then we don't need to do any of these steps here. But to measure that volume, we can just go in and use this separate tool. So we would just say, other tools, volume measurement, create an image for the volume measurement. Let me just select that folder again. Select a channel. And I can just give it a name. This will just create like a slightly scaled down image and save it in the folder here. There'll be a folder called threshold measurement that is in here. It'll have so actually I just ran this earlier, but so let me just get rid of this. It'll include the channel name, so it's channel 1 and then what the name that I gave it. And then I can sort of look through this data set. Actually, I'll show you in a second, like you might want to make some changes to this image to make it easier to make a measure. Maybe like there's some bright signal here that I don't want. Like maybe I really just want to measure this stroke and I don't want any sort of other information to be detected. Actually, I can even show you how we can do that live. So now that I have this data set, I just run the next part of the pipeline. We need to just tell it, okay, which data set are we analyzing? It's this one here. The image that we're going to measure the volume. So this will be our threshold measurement. Here's our data set here. The atlas we want to use, maybe we want to use the Kim atlas, maybe we want to use the Allen Brain atlas. So you would select that here. And then the Ilastik directory. These are all these fields, they auto-populate based on the previous thing you've done. So, this should always be correct once you've set it. This you'll have to change only if you change the atlas. So you can save a little bit of time there. Okay, so it's going to open up the image and it's going to open up the threshold tool as well. And this gives you an opportunity just to like, play around with the threshold first. And you could also do something like, okay, like I can see that there's definitely some stuff that's not the stroke that I don't want to measure. So this, you could use this opportunity to actually just go through, I could just go, I can just clear outside. So now I've got just this stroke area that's been left. Adjust my threshold a little bit better. And then it's tempting to click apply here, but you just want to click OK. And then it's going to do that threshold. It'll make you an image that actually is mapped with the atlas, so you can visualize those brain regions that the stroke is in. And then this will take a couple of minutes. This is doing the reverse mapping and region analysis. Maybe while it's doing that, we can actually have a quick look at the image. So this is actually like a raw data, but now it's actually mapped into those brain regions. And then in a minute, it's going to give us the table. Actually, while we're waiting for it to do this, I can show you the sort of final example, which requires a little bit more like playing around on the image to do. So here, this is a good example where we have like a GRIN lens that's been inserted into the brain, and you might want to measure, where the lens is, but you might also want to know, like, okay, where was I imaging exactly in the brain? And so this is a data set that Anej was able to share with me, and we can look in here, and so I'm just going to show you. And we can see this pretty clearly on all the channels because there's a big chunk of the brain missing. Yeah, so here, these are like pretty coarse sections. This was like, these are 75 micron sections, I think, but it's every third section. But we were still able to process it in BrainJ. So, it's obviously better to have thinner sections in terms of having better resolution in the anterior to posterior axis. But if you are doing like, sub-series of, to analyze different probes and things like that, it's still possible to do the analysis with BrainJ. So you can see here, this is like the placement of the lens. It's kind of placed on a bit of an angle, so you can see this is kind of like where we're mainly imaging here, and then it starts to disappear. So if you wanted to measure this, you could do two things. You could, we would have to sort of create the image to analyze. So we do the same step as I showed you before. You would go to plugins, BrainJ, volume measurement, and create the volume. And then I'm just going to show you here what you would do. So after you created those images, you could then start to just, very quickly, you would just manually annotate, where the lens was or where the imaging area is. And you could use like a polygon or something like that to easily draw that out. So, you can just draw on the image this way. And an easy way to do this, I'll just do this very quickly, but you could spend more time and do it more accurately. When you draw a region like this, if you press F to fill it, it's going to fill with whatever the intensity is on the maximum over here. So just keep that in mind. Filling it with this 4000 is not so good because there's other signal here that's actually 4000. So a better thing to do would be, if I look at this with the stretched out, okay, so it's finished here. Let's move this out of the way. If I just stretch this back out to the full range, I can kind of still see the tissue, but it's a little bit difficult. This is kind of where I would use something like a lookup table to make my life easier. So, a very common one is to just use fire. Now I can see the tissue very clearly, and I can then just, fill this with a very bright value. And then, I can move from slice to slice. I won't, bore you with doing this for every section, but, I can modify this a little bit, fill it, go to the next section, adjust a little bit more, fill, and then basically I'd have an image that's annotated for this lens as well, and I could do the same thing for the imaging area. And if I look in here, this is where I've actually done this already. So here I've got like the GRIN lens. I'll just quickly show you. So you can see where I've done this, and also kind of looked at what, like the 300 micron sort of depth that we get using these lenses. This is just an approximation just to demonstrate. And then we can then run the analysis as I showed you. We'll get the same images that have the thresholded, let me just, so you'll have an image that shows you like what brain regions you were imaging in. So this was actually analyzed in the Kim atlas. So these are kind of the different regions in the striatum where the lens was placed. And then you'll get a measurement here. So this table will then show you what the volume is for that brain region, and then what the volume is of, let me sort this so you can actually see. Yeah, so there's a very small volume of this lens, like imaging area. So I don't know why it's not sorting properly, but that's you know, how you can measure a brain region, and hopefully that's clear in here as well. This is our stroke, and we can see it's mostly in the left hemisphere here. Yeah, I think that's like the main things I wanted to show you about how to use the pipeline, and hopefully like that was also useful in terms of how to use Ilastik as well. You know, if you're interested in learning more about that, then, you know, please come to the Ilastik workshop. We'll go into that in more detail and just how to build that into a pipeline in ImageJ if you want to do, you know, like a different project that takes advantage of that and automates it. And I'd say also, like, definitely reach out to us if you want to start using the deep learning on the spinning disk or the slide scanner, because I think, you know, that's really the way forward, is using that segmentation at the microscope. You know, it's like just a few seconds of processing time after it takes the image, and then you have the locations of the cells, all the axons and dendrites, and then you can quickly just go through and do the analysis side becomes very easy then, because you've done the hard work at the microscope. And I think the results, you know, are potentially a lot better because you can have a lot more robust and reliable segmentation using those tools if it's trained well. So, is there any, you know, questions or you know, anything you want to ask?
Yeah, I have a few questions. You know, one was, how can it deal with a hemi section if you just have half brain, or do you need to duplicate it and flip it and recreate a full section?
No, if it's a hemi section, then like the way to deal with it would be to, I would probably just create a, go into the Atlas folder and actually just remove the hemisphere. And that way, like, yeah, it might take a little bit of troubleshooting. Like, you know, I can have a, you know, if you're having trouble with that and that doesn't work immediately, let me know and maybe we can figure something out. But I would say, like, as long as you've got the hemisphere sections and they're, you know, in good condition, and you modify the Atlas in that way, you should be okay.
I see. Yeah, and the other thing I was wondering, so what is the machine learning on the computer? And like something I've used that's been really good for neuronal segmentation is cell pose. And I was wondering how easy it is to just integrate that instead of Ilastik to make for a set of masks.
Oh yeah, that's a good point. Yeah, so yeah, cell pose is great. And yeah, I can have a look. It shouldn't be too difficult to like support that. Yeah. Yeah, and in terms of like on the microscope, they don't share the architecture of what they're doing, but it's basically like a convnet for, you know, image segmentation, like a UNet, most likely. But it works, you know, really well. Like we've been able to train it already quite well, and I think with some effort, you know, I think it's going to be better than using Ilastik, and it's faster. I think I didn't mention, but Ilastik is about 20 seconds per channel per image. Which isn't bad, but like if you have, you know, 300 sections, that's, you know, a bit of time. It's like 2 to 3 seconds, I think, for the deep learning segmentation. So it's much faster and better.
Yeah, and then the last thing is, I have some anterograde tracers that use synaptic markers. So you're getting like dots, and is wondering what the best way would be to use that as the projection information.
Yeah, so I mean, I guess if you can really like identify, if you've got enough resolution to see them as discrete like points, then you could almost treat it like a cell analysis.
Yeah.
Yeah, and if you were to do that and you had to have more resolution, it's definitely going to be a bit like, it's going to take longer for processing, but yeah, it could be treated as a cell analysis problem.
Yeah. Okay. Yeah, that was all. Thanks.
Yeah, no worries.
Luke, I have a question about the registration in terms of results. So I understand the part, of course, if you have damage on the slice that, you know, actually, I think you know that I use a lot whole brain. I'm using still whole brain as software for doing that. And so also I wanted to say for this thing, when I have problem, because maybe there is an asymmetry also from brain cutting, I actually now mirror it. So I slice the brain like in a hemisphere and then I mirror. And so I'm gonna use the same atlas reference, but I know that it's just 1/2 actually that I'm gonna consider them for counting. But that's just, you know, practice. But there, what I like with that software, I haven't used yours yet, but I already saw it, so I'm gonna actually try it, is the fact that the registration can be adjusted. So on single image, if I see that the ventricle, for example, really the matching was no good, I can manually adjust the points. Is it possible to do it in the registration that is in your plugin or not?
No, so manual adjustment is not possible. So it's really, I mean, but that's kind of, if you have good quality data, the registration will actually be very good. And even if there's damage, the registration should be also good, but you just have to look at, it'll be like, the issue will be like a local area. Like if you have like some stuff, you know, missing from the brain, in the registration, the cells in that sort of local area will be kind of poor, but you should overall like, I mean, I think even the sort of spinal cord data sets show this actually quite well. I mean, it's kind of hard, these registration problems, if there's no landmarks to help the registration, you would think that things could be, the accuracy could be quite poor, but like, that's the case for a lot of the spinal cord. There's really just the gray matter and the white matter. There's other regions that are a little bit difficult to sort of define with neurotrace, but we can still get good results. And it's, you know, same with the brain. So yeah, you shouldn't have to do any sort of manual adjustments afterwards. But it does depend on the sections being good. If the sections are really badly damaged, the pipeline is not so useful.
Yeah, that makes sense. Thanks.
Yeah, no worries. Yeah, well, thanks for coming, everyone. And yeah, I'll like edit this and you know, share it hopefully in a week or two. And yeah, just reach out if you have any other questions or anything like that. Yeah, thank you.